Hello, Year 10s. So far, we've looked at 4 factors that affect the rate of the reaction. We have looked at the temperature. Increasing the temperature speeds up the reaction. Increasing the concentration, increasing the surface area by crushing it into a powder, and adding a catalyst. And it's that area that we're going to focus on today. We have also had a look at different methods we can use to investigate the rate of a reaction. We've had a look at analyzing graphs from the results of rates of reaction, and also how to make calculations to calculate the specific rate of a reaction over time or at a specific point on a graph. So let's focus on catalysts. And I know at times you say, what's the point? Why are we doing this? How does it apply to us? Well, hopefully this picture gives you lots of examples. So the first one I'm going to concentrate on is a group of catalysts that are found in living things. They're not actually alive, but they're found in living things, so they're called biological catalysts or enzymes. There's an enzyme in the fungus yeast which helps to speed up the chemical reactions that makes wine and beer and bread. So the yeast works on the sugars found inside the hops and ferments it, turning it into an alcohol called ethanol, and carbon dioxide is given off at the same time, which creates the bubbles. In wine, the grapes are fermented to produce the alcohol called ethanol. And in bread, in fact, sometimes in the oven, you can smell that yeasty alcohol type smell. That is, in fact, the ethanol produced in the reaction, which is evaporated, which is why we don't taste it in the bread. And the carbon dioxide bubbles that's produced in that reaction produces the holes that we see in the bread and helps it to rise. So, a very, very important enzyme found in yeast, catalyst found inside yeast, that speeds up this chemical reaction to make these very useful products. Baby food. Now, as you can imagine, a baby's digestive tract isn't mature enough to break down all the foods that it eats. And that's why, to help support their digestion, manufacturers add enzymes inside baby food to help start to break it down, to start to digest it, so that it's easier to move across the wall of the small intestine of the baby, so that the food can travel into their bloodstream. I wonder if you knew about that. And then I've got this environmentally friendly washing powder called Ecova, and it is a biological washing powder. Now, the reason why it's called a biological washing powder is because it has enzymes in it, and these enzymes help to break down any proteins or carbohydrates or fats that you happen to spill onto your shirt if you're being a bit of a mucky puppy. And the enzymes help to speed up the reaction, to break it down, so that when it comes out the wash, you can't see the stain on your clothes anymore. Ever wondered how come you can get cloudy apple juice and clear apple juice? That is due to the work of another catalyst, which is an enzyme called pectinase. What a cool name, pectinase. And again, all enzymes tend to end in the word ase, or the letters ase, pectinase. So pectinase breaks down the cloudy bits in your apple juice, leaving it all lovely and clear, if that is your preference. And finally, the last picture is an application we have learned about a catalyst before, known as the Haber process. So in the Haber process, hydrogen and nitrogen combine in a chemical reaction to produce ammonia. Now, if you remember, ammonia is very important in producing fertilizers which help to grow the crops that feed all of us on this amazing planet. And this reaction is sped up by adding a catalyst, which also lowers the energy costs needed to help this chemical reaction on. So a very, very important application and a bit of revision of your biology and chemistry. So let's focus on why or how catalysts exactly work. Okay, so to start off with, we remember that a catalyst is very often a transition metal. In biology, we call them enzymes that increase the rate of the reaction. Increasing the rate of the reaction is the light bulb that needs to go off in your brain when you hear the word catalysts. How do they do this? They provide an alternate pathway for the chemical reaction by lowering the activation energy of the reaction. Remember, the activation energy is the energy needed to get started. The next fact about catalysts, they don't get used up in a chemical reaction, and we're going to talk more about why that is so useful. We're going to have a look at some examples in industry, some examples in living things, and very, very important, the GCSE syllabus requires us to know how to make beer and wine. So let's have a look at these in a bit more detail. This graph shows us what happens when we add a catalyst to a chemical reaction. The graph compares the energy of the reactants with the energy of the products. And in this reaction, the reactants have more energy than the products. But in order for the reaction to get going, the blue graph at the tip, that is the amount of energy needed before the reaction can get going. That is known as the activation energy, and it's measured from the height of the reactants to the top of that curve. Now you can see with the red graph, by adding a catalyst, the activation energy has significantly decreased, and that is why the rate of the reaction will be a lot quicker. It's really important that you know this graph, that you know what the activation energy is, and that you can show which one is the graph using the catalyst. Now this is a picture of the exhaust pipe from a car and a catalytic converter. We know that in catalytic converters they use some very, very expensive transition metals such as platinum and palladium. And what they do is they help to speed up a reaction to convert some of the harmful exhaust gases into harmless gases to stop harming our atmosphere. And the reason why they can justify using such an expensive catalyst is because the catalyst gets used again and again and again. It doesn't become part of the reaction. It's the same at the start of the reaction as it is at the end of the reaction. And that's a very, very significant point to remember. Now, the example in industry, which we study in our GCSE syllabus in a lot of detail, is the Haber process. As I said, the nitrogen from the air reacts with hydrogen from hydrocarbons and gets turned into ammonia. This helps to save money by lowering the activation needed for the reaction to take place. So I don't need to add in a lot of energy to get the temperature high enough to get the reactants to start reacting with enough energy. Notice how I've written this reaction. I've put iron on top of the arrow rather than as part of the reactants and part of the products. This is to show that it doesn't actually change during the chemical reaction. It simply acts as a catalyst, speeding it up. Now, we're going to focus on enzymes in living things, and this really is simply revision of some biology work you've done. We know that the catalysts found in living things are called biological catalysts or enzymes. And there's so many different uses of enzymes, from respiration to photosynthesis to the digestive system. We know that enzymes are protein molecules, and they've got a specific shape because of their active site. A little bit like a lock has a specific shape that only a certain key with the matching shape can open up. So because of that active site, because of that specific shape, they only work with very, very specific substrates. So for example, when I eat bread, which is made out of starch, the enzyme in my saliva known as amylase, it can only break down starch, and it turns starch into glucose over time. And that's why if you put a piece of bread in your mouth and leave it there for a little while, it will start to taste sweet. If you haven't done that before, give it a go. So amylase will only work on starch. It won't work on a protein or a lipid at all. We also know that enzymes are fussy. They work best at an optimum pH or temperature. And that also means that if I go above that temperature, above that optimum temperature, the enzymes start to break down because there's so much energy that the shape starts to fall apart, which means that the active site becomes destroyed. Now be careful, because a lot of people make the mistake of falling into the trap and saying the enzyme is killed by the high temperature. It's not killed. It's a chemical. It's not a living thing. The key word is it is denatured. It's denatured. And finally, making beer and wine. What do you need to know? You need to know that the enzyme in yeast breaks down the sugars that's found in beer, or sorry, that's found in hops and it's found in grapes, called glucose, and it converts it into alcohol, which we call ethanol, and also carbon dioxide gas. And we know it's carbon dioxide gas because it turns lime water cloudy. And here is the chemical reaction for that. So glucose gets broken down into ethanol plus carbon dioxide. Now, I want you to stop at the screen. This is where I'm going to end now. And this is the summary that I'd like you to create. Use a whole page in your book. Do it as a mind map. These are the key points you need to know about catalysts. And then you can take the quiz.